Okay, good morning, everyone. So we're going to start. Welcome to the seminar. So as usual, we are on a webinar mode. So people online, do not hesitate to report any technical issue in the chat. And please ask your question rather in the Q&A tab, and we will allow you to interact with the room during the discussion session. And with that, thanks very much, Anne-Geneviève, for introducing to the speaker.
Hello, good morning. So I'm very happy to present you Joël. So I know Joël since a long time, and first of all, he was working on ants, and he has done his PhD with Michel Chapuisat in Lausanne in Switzerland. And then he went to Bâle to work on earwig. So his new model since then in 2012. And then after that, he moved to get an professor position in Germany. And finally, he arrived to get a position at the CNRS in 2016 at the place where I was at the time, at the IRBI, which is the Research Institute working only on biology of insects. And unfortunately, I left in 2017 to be here at the CEF. But that's why I invite Joël, because first of all, in fact, he comes from Montpellier, so he's doing the cycle. Thank you, Joël.
Thanks a lot, Anne-Geneviève. Thanks a lot for the invitation. So as you said, actually, I was born in Montpellier, so I know the city quite well. But I have to say the last time I came on the campus was maybe 20 years ago. And I have to say it didn't change that much in terms of building. So I somehow recognize the place. Anyway, so I'm very happy to be here and to be able to give you an overview of one line of research we are doing in my group, or we have been doing over the last maybe 10 years, which is about social evolution in insects and how microorganisms could be linked to this social evolution in this kind of organisms. And so social life is clearly a ubiquitous phenomenon in nature. You can find it in almost all animal taxa, from vertebrates to arthropods, which includes, of course, insects, but you can also have it in arachnids and many other invertebrate species. And one thing, however, is that even if it's present everywhere, it's not the only way of life. So you still have quite a lot of species that are solitary. So a major question is basically why we do have species that live in groups and others that remain solitary, or the other way around. And this is particularly of a problem because when you live in a group, in general, you have a high risk of pathogens. And this is basically due to three main factors. The first one is that, as the COVID-19 taught us, basically there is a high risk when you live in a group to transmit the pathogens to other group members or to receive the pathogens from other group members. The second main issue is that in general, when you live in a group in nature, you have a relatively high genetic relatedness between group members. That means that if there is a genetic, somehow a genetic weakness, this weakness will be shared among all group members, and the same kind of pathogens can come and kill all group members or affect all group members at the same time. The third problem is that when you live in a group, in general, you also have a high nest fidelity, and you can build a nest or modify the environment to improve the development of your offspring, but the same condition may improve the development of microorganisms in the environment. So basically, if we focus on the three main issues, we should not have group living in nature. Nevertheless, we do have group living in nature. And so one of the major questions is basically how group living did manage to evolve despite this inherently high risk of pathogens in the environment. And well, the answer to this question is based on, again, two pillars. The first one is relying on the individual immunity, so the immunity that everyone has, which is also called the personal immunity, and which typically involves physiological immunity, like basically the immune system, and behavioral immunity, where you're going to avoid to go to infected places, for example. But when you live in a group, you also have access to a second layer of defense. And the second layer is called the social immunity, which is basically when group members can help you to improve your defense against the pathogens. And this concept of social immunity has been developed, let's say, 15 years ago, and mostly in eusocial insects, where, I don't know, okay, where the different forms of social immunity have been described. So you have allogrooming, where one individual will groom another individual to remove external pathogens. You have in some termites, for example, you have alarm behaviors, where some animals going to shake the body when there is a risk of pathogen infection. You have the use of natural components with kind of self-medication, with the use of resin in some ant species, where they bring back the resin into the colony, and this resin has some antimicrobial property that's going to protect group members from the inside. And you have sanitary and hygienic behaviors, where basically the animals going to remove corpses or infected animals from the rest of the colony. So as I said, all these examples have been mostly described in eusocial species, so that at some point it was trust that social immunity is something that is very specific to eusocial species, and that is something that has secondarily evolved because the animals did not have, had to live in groups. They didn't have the possibility to live by themselves and to escape from this risk. But the problem is that, the question remained open at some point because nobody really investigated the presence of social immunity in non-eusocial insects. And so an alternative hypothesis would have been that social immunity is actually the driver of sociality by itself, because it allows solitary animals to get access to an additional layer of defense against the pathogens. So is it something that is specific to eusocial species, or is it something that comes with the evolution of group living per se? And as I said, the main issue is that when you think about social evolution, you have a long transition between solitary and eusocial species, and you have a lot of different stages and different forms of social life. And to date, most of the knowledge about social immunity is focused on this eusocial system, which is very entangled, very derived, and there is not so much information about whether it's present or whether we do find some of these forms along the evolution of social life. And there is one particular step that is very important to focus on, which is at the very beginning of the transition between solitary and group living, which is about species that are gregarious, so they live in groups, and they do have a family life where you have parental care. So these two things are the first steps in the evolution of social life. And what is also important is to focus on species where these two steps are facultative. So that means at any time in their life cycle, they have the possibility to live by themselves or to live in a group. And there we can test whether they have benefits of living in a group to have access to social immunity. And this is basically what I have investigated over the last 10 years, mostly focusing on this species, which is the European earwig, Forficula auricularia, that you probably know. Here, this is a female, this is a male. You can recognize the sex based on the shape of the forceps. So the female have straight forceps and the males have curved forceps. At least you've learned something today. And what is interesting in this species is that they do have a social life. First, there is forms of social interaction in terms of maternal egg care. So the female starts to lay eggs at the beginning of winter, and then all of her winter, for about 2 months, she will stop feeding, stop doing anything else than providing care to the eggs. And these forms of maternal care is obligatory. If you remove the mother, the egg's gonna die. After egg production, after egg care, then the eggs will hatch, and the female will remain with the offspring for about 2 weeks. And during this period, she will provide a lot of care, she will provide them with food, but these forms of family life is facultative. So if you remove the mother, the offspring are able to forage for themselves and basically can survive very well without a mother. And then you have the adult stage where they do form groups. They are highly gregarious. You can have groups of up to 300 or 400 animals. And this is basically the rest of their life cycle is about this group living. But here again, this group living is facultative, so they can basically live by themselves. So here we have a species where we do have family life, the gregarious form of life, and all of them are facultative. So every animal at any point in its life cycle has the possibility to live by themselves or to join a group. So we did investigate whether living in a group of having access to maternal care indeed provides some benefits to the offspring or to the animals in general. And for example, here, so in principle, this is a video, it's supposed to be moving, but here you have a mother and here you have the eggs here. And what we have found is that if we indeed cover the eggs with spores or fungus, then the mother will increase her grooming activity and clearly remove the eggs, the spores from the eggs. So basically for the offspring in terms of eggs, there's a clear benefits against pathogens of having a mother around. Then what we found also is that the way the female build a nest may also depend on the presence of pathogens. And here you have two petri dishes where you have sand. Here you have the female with the eggs, and here is the same. The female is just smaller with the eggs as well. And here you see the female dug into the sand and actually build a quite sophisticated nest. Whereas here, if you manage to see it, the female only produced the eggs on the ground. So she didn't actually build anything. And what we found out is that this is actually driven by the presence of pathogens in the environment, which is not the case here. So if there is a risk of pathogens infection for the eggs in the environment, not on the eggs, but just in the environment, then the mother will is more likely to build a sophisticated nest. And the last thing we have shown, last example here is that, so we have the offspring here. So this is again a petri dish. You have the offspring, and what you see, which is shown by the red arrows, are actually their feces. So they are living on their feces. And what we found out is that these feces have antimicrobial properties, so that basically the mother and the offspring keep the feces into the nest, lay the ground of the nest with their own feces to basically protect or prevent the growth of different types of microbes in the nest. So they can sanitize the nest with their own feces, which is not something to do at home. But then the question we had is, okay, all these were short-term benefits. So clearly benefits of maternal care in a very short period. Do mother can provide care or benefits against pathogen infection over the long term? And so one thing we recently explored is whether this post-oviposition care could actually allow the mother to transfer some immune factors to the offspring for the future development. And this is based on the recent development of knowledge about the acquired immunity in invertebrates, where it has been shown now there is quite a lot of papers that shows that if you infect of the immune challenge a female or even a male, and then you wait a bit, then this animal will develop a kind of protection that can be specific or not specific to the pathogen they have been exposed before. And there is a form of acquired immunity that has been shown. And then there are all the papers that show that this acquired immunity can be transferred to the next generation. So if the female is primed with the pathogen before oviposition, then she will transfer something into the eggs. And when the eggs will hatch, they will produce new adults that get the same protections. So having a mother that is challenged before egg production actually is beneficial for the transmission of the protection to the descendant. The problem, this is called the transgenerational immune priming, and it has been explored in quite some invertebrate species. So here you have a phylogeny that shows different groups of invertebrates. When you see they are between squares, that means that the presence of transgenerational immune priming has been investigated. Here you have the number of species in each order where it has been investigated. And when it's gray, it's the proportion in species where it has been found. So here, where it has been investigated in one species which has TGIP, here in Diptera, it has been investigated in 5 species, but only one had TGIP. Just for you to know, earwigs are here. These are the Diptera, and at that time we had no knowledge about the TGIP. So the thing with TGIP is that it's very efficient. It has been shown to be present in many species, but there is a major issue, which is that it implies that there is not so much mismatch between the pathogens that the mother will encounter before oviposition and the pathogen that the offspring of the next generation will encounter when they become adults. And well, that might be the case, but here our hypothesis was that maybe this mismatch could be somehow corrected by having parental care. So if a mother or father that provides care to the offspring or to the eggs is contacting the pathogens while they provide care. If they manage to transfer something, then they will reduce the distance and the risk of mismatch between the 2 points. The problem is that whether parental care can mediate this transfer of immunity has actually never been investigated in vertebrates. So, but surprise, surprise, this is what we decided to investigate in earwigs. And so the first step was to know whether we do have forms of immune priming. So first of all, we want to know whether actually there is something that could be transferred. So are females that are primed better resistance against an infection later on? So what we did is we had 4 treatments. So females that were providing care. So all these females were taken 47 days after egg laying. So the eggs are out, the female is taking care of the eggs, and then we take the female, and either we inject the female with a high level of dead bacteria. So this is the bacteria we used. So high concentration of dead bacteria, which is a strong immune challenge, a low concentration of the dead bacteria, which is a mild immune challenge, or we injected with PBS, which is used as a control, just for control for the effect of wounding. And then we had the pure control where we did not do anything to the mother. After that, we wait 7 days, and then we injected all the females with a live bacteria, concentration of bacteria with LD50, so which is supposed to kill all of the females in about 21 days. And then we checked whether the treatment had an effect on their resistance. So this is what we have here. On the bottom, we have the days after injection, so over 21 days. And here, the cumulative survival rate of the females between 0 and 1 means all the females are alive. This is what we have for the control. So the female that we did not manipulate it at all. And the good point is that we are about 50% of survival or death after 21 days, which was supposed to be our lethal concentration. So that was great. Now, what was more surprising is that this is the survival of the female that were injected, so primed with the highest concentration of dead bacteria. And what we see is that these females actually survive less well than the control females once they are injected with live bacteria. So that means that there is a kind of cost of immune challenge. So you first activate the immune challenge with a high concentration of bacteria. There is a cost of activating this immunity. And then when we inject a lethal bacteria, then the cost is already too high, and then the female will die earlier. This is what we have. Now, what we have with the low concentration of bacteria is basically no different with the control. So it's really linked to the threshold at which we activate the immunity. What was more surprising is the result we have with the PBS injection. And with the PBS injection, which is, as I said, a control just for the effect of wounding, with the PBS injection, we do have an immune enhancement. So that means if we have a female, we inject them, or we just wound the female, and 7 weeks later, we inject them with a live bacteria, then this female will survive better than if we didn't do anything with the female before, or if we injected the female with bacteria as well. So, well, the good point is that there is something going on. There is an immune enhancement. So when we do something to the female, it affects whether the female is more or less likely to survive to an infection. What was more surprising is that we had this effect with PBS and not with dead bacteria. And so this raised some questions. The first one is what kind of immunity or immune response is triggered by the PBS? Is it a sustained immunity? So that means we harm the female, she activates her immune system, which is stay high for 7 days. And then when we inject the female with living bacteria, then it will be already activated. And so it will be very efficient. Or is it a recalled immune response? So it's activated with the wounding, and then it decreases, and then it's reactivated and more efficient once we have the pathogen, the live pathogens. The second big question is about the fact that this is about with PBS. So it's not a specific immune challenge, it's a non-specific immune challenge. And what is a bit surprising is that it somehow suggests that this that the specific immune challenge nullify the effect of the wounding, because in these two scenarios, we also have PBS. So it's bacteria plus PBS. So having the bacteria nullify these benefits. And this is a kind of question. And the largest question is that, again, it's a PBS. So if it's wounding, it's very non-specific. It is very frequent, at least in insects. And so it raises questions about the evolution of such a system, because we could expect that almost all animals at some point will be wounded. And so how this could be maintained over time and over generation. And well, these are just all non-answered questions. Nevertheless, we had a form of not immune priming, but at least immune enhancement with the PBS. And so the next question was to know whether the female can actually transfer this immune enhancement to the next generation via maternal care. To test this, we had exactly the same setup, except that this time, after we exposed the female to the different treatments, we let the eggs develop, which takes about 2 months. And then we have new adults. Then in these adults, we checked many life history traits. So for example, the clutch failure here, how many eggs managed to hatch, the developmental time of the offspring, the nymph weight, the survival rate of the adults, etc. None of these traits was associated with the treatment. Great. So that means then we can test really what we want. So we have the adults, and about 1 month after they became adults, we did inject them with a lethal concentration of the pathogens, and then checked whether the treatment of their mother while she was providing care actually increased or decreased their survival. And what we found is more or less the same pattern. Here we have no difference between the control and the offspring from mother that were injected with dead bacteria. But again, from mothers that were injected with PBS actually had a higher survival rate. So that's great, because it shows that there is this immune enhancement can be transferred to the next generation. And that shows for the first time that it can occur via maternal care after oviposition. So this is a process that can really help the offspring once the eggs have been hatched. Whether this is egg care, whether this is nymph care, or both, is something that we are now investigating, because, well, in our first experiment, the mother had access to the 2 types of care. What is interesting here is that it shed light on the new benefit of care for the offspring. And this is kind of important because actually over the last decades, we accumulated a number of data that shows that in earwigs, having a mother is not necessarily very good. In different conditions, actually the offspring survive less well if the mother is around, or if there is bad conditions, the mother is actually decreasing the care than increasing it. So here, maybe we have something that may explain, at least in part, why the juveniles have some benefits in remaining with a family and with a mother living and adopting a solitary life. The last question is, of course, what is transmitted? We have the PBS is doing something, but what the mother does transmit to the next generation? Is it about antimicrobial peptides, hemocytes, or other molecules, or cellular promoters of immunity? This is also something we don't know. So this, up to now, I've only talked about pathogens, but microorganism, as you all know, is not only about pathogens. It's also about some beneficial microbes and symbionts. And there is, among them, of course, there is a big rush on the gut microbiota, so this community of microbes that live in the gut of the host. And now we know that they are able to affect lots of life history traits in the host. There could be immunity, physiology, reproduction, behavior, many different traits. And recently, it has been suggested that there is a link between this gut microbiota and social evolution as well. So the classical theory of social evolution are kin selection, reciprocity, multi-level selection. All of them are focused on the benefits of group living for a single individual. So how they did get direct or indirect benefits of interacting with other group members or being in a group. The thing is, this novel hypothesis is actually very independent of this one and suggests that group living may evolve due to gut microbial manipulation. And this is because first we have an accumulation of data now that shows that with the gut-brain axis, that basically all the microbes we have in the gut are able to manipulate the host. So are able to modify many different things in the brain of the host and the behavior and all these points. There are also some data that shows that, particularly in the context of parasitism, that some microorganisms are able to manipulate the behavior of the host in a way that improves their own fitness and not the fitness of the host. And here you have, for example, this classical example of fungi manipulation in ants. So if you put the two things together, it might make sense to think that the gut microbes might manipulate the social life of the host to make them more social. And by doing this, increasing their own transmission to a new host. In this way, that would mean that social evolution or the evolution of group living could emerge independent of the benefits of group living for the host itself, but only for the benefits of the gut microbes that is present in the host. Well, this is a very tricky scenario. It has been investigated a bit in the last few years in different insects, cockroaches, locusts, termites, honeybees, and ants, where basically what the research did is to provide them with antibiotics or to form selection lines with axenic lines, so lines without gut microbiota, and see whether the alteration of the gut microbiota induce a reduction in the expression of social behaviors. And they did what they found, for example, in cockroaches and locusts, is that the animals that get antibiotic, less gregarious or attract less conspecifics, they may be more aggressive, they have social interaction and social networks that becomes completely crazy or less efficient, and they may also increase aggressiveness in different contexts. The thing is that all of this shows that gut microbiota alterations makes social individuals less social. It does not answer the opposite question, which is whether gut microbiota make solitary or poorly social animals more social, which is the hypothesis I was interested in. So, surprise, surprise, again, we tested this with earwigs. Another picture just to show you that they do have wings, because nobody knows that. Why we use earwigs, and in particular adult earwigs, because they do form group, as I said earlier, up to 100 individuals. And there were some reports in the literature that says that some animals are more gregarious than others. So we had a hint about the presence of natural variation in whether animals are solitary or not. And we know that in these aggregations, they do express a lot of behaviors that could allow the transfer of gut microbes from one host to another, which is mouse-to-mouse contact, allogamy, allocoprophagy, etc. So what we decided to do is to process in 3 steps. The first one, we wanted to quantify this natural variation in aggregation level, just to make sure that indeed we have some animals that are more social than others. The second step then would be to try to see whether we have a link between this natural level of sociality and the composition of the gut microbiota. And the third step was to know whether we could manipulate this gut microbiota to make individuals more social. So for the first step, we went to the field. We collected more than 1,000 females. Then we bring them back in the lab and we put them up in a setup like this, where we have cameras and all animals are in arenas, which look like this. And in these arenas, we had 4 cells. In one cell, we have a test female that was free to move all along the 3 cells. And on the top cell, there were 3 stimuli females that were actually connected to this cell with a kind of tunnel that allows antennal contact and visual cues. But these animals were too large to go through this, and this one to go through that. So we set up all these groups of female in this kind of setup. And then we take a picture every hour for 72 hours, so for 3 days. And then we checked on these pictures whether the test females was here or not. And basically from this, we extracted an aggregation scores from 0 to 72. 72 means the female was always near conspecifics for the 3 days, for every hour. And 0 means she was never, so she was either here or here. So this is what we have here, these aggregation scores from 0 to 72, and here the number of females in each of these scores. And this is the result. So as you see, well, there are some variations. These variations are non-normal and non-uniform distribution. Here is expect from a random distribution, and here is another expectation from a random distribution. So something is going on. But the main question was still to know, okay, we have a scores, but does it make sense? So what we did is we extracted the score per day, and we checked whether what we recorded on the first day was correlated with what we observed in the second day and the third day, and etc. So we tried to see whether there is some consistencies in the scores we recorded. And lucky us, this is exactly what we had. So even if there's lots of points everywhere, if you look at here, we do have correlations between the aggregation scores from one day to another, whatever the day is. And here you do have the sampling weeks, because this was a setup that allowed us to film 66 females per week. So we had to repeat it for 5 weeks to reach a large enough number of replicates. So here what we have is we just confirm what was reported in the literature, that indeed we do have some natural variation in whether females are more or less social. So then what we did is we tried to see whether they do have a different microbiota. So from this distribution, we selected the most gregarious female and the least gregarious female. We dissected them, extracted their guts, and using bioinformatic pipelines, we actually ended up with microbial taxa that were present in their gut. So we used ISV sequences. And this is what we have. So here we have all the females with the low aggregation scores, so the least gregarious females. And here you have, well, the distribution of the gut microbiota. So each color is a different microbial family. And well, you see the blue and different colors. So the main question was, what do we have with the most gregarious females? Do they have something that is completely different, or is it completely the same? Well, second option. Unfortunately, it's not very obvious. There is not something that is clearly obvious about the fact that most and the least gregarious females have different gut microbiota. And it's not only a matter of observation. So if we go into the calculation of the different indices well, it confirms the first impression that clearly there is no possibility for us to disentangle whether a female is highly gregarious or poorly gregarious on the gut microbial community. But the question was not only about the community, it was maybe about the presence of certain microbes that could make them more or less social. So then we checked at the sequences, and if we use a sequence, we do indeed find a total of 10 sequences that should be able to discriminate between the two categories. So we were very happy. But the problem is that that was about the sequences. Now, if we check at the individual level, so the host level, here you have the different sequences that were supposed to be discriminant. And here you have the 20 females with the low aggregation, 20 females with the high aggregation. And here you have the number of females that carry these sequences. And as you see, this is not very convincing. So basically there is no sequences that are only carried by 20 females in one category and not the other. So in terms of sequence, that just means that in the entire pool, a few females contributed a lot in the number of sequences that, well, basically covered the entire pool, but it was not very convincing at the host level. So, well, we did not manage to really shed light on the link between that. But nevertheless, we wanted to see whether we could manipulate it, because here, analyses were based on the bacteria. And maybe in the gut microbiota, there is other parameters, other components that could influence anything. So what we did is we tried to manipulate this gut microbiota. So here again, we selected females that were poorly gregarious or highly gregarious. And we dissected them, we extracted their guts, and we give their gut to feed to females that were poorly gregarious. This is a species that is highly, highly cannibalistic. So it's very easy. You extract the guts, you give it to a female, and she will eat it immediately. And so we have poorly gregarious females that were fed with guts from poorly gregarious females, and poorly gregarious females that were fed with guts from highly gregarious females. So our expectation here, well, so we gave them feed, and then we measure the aggregation scores, so the change in aggregation scores between the two processes. Here, there were our predictions. So this is the change in aggregation scores, 0 meaning there is no difference before and after. Positive values mean that they become more gregarious, and negative values that they became less gregarious. That were our predictions, and this is what we found. Well, nothing. So basically, here what we show is that the origin of the gut transplant actually do not modify whether a female will become more or less gregarious. It seems to be completely independent. What was more surprising is that if you look at the two scenarios, well, the gut transplanted females, all of them became more gregarious, which doesn't make sense, at least according to our hypothesis. And so this is a kind of a big question. And for that, we have different interpretations. The first one is there is temporal variation in aggregation scores. So the life cycle of the earwigs is that at the beginning of summer, the new adults and new females are highly gregarious. And at the end of the year, they become more and more solitary because then they will have to produce their own eggs and they become solitary to lay the eggs. And so maybe here, what we have is we have something like a transition, seasonal transition in whether or not they are gregarious, which is completely independent to the manipulation we've done. The other question is about the impact of the social environment on the aggregation scores, because we always tested them with the same conspecifics, and maybe this, the level of aggregation is driven by the conspecifics and not by the host itself. So no effect or no link between gut microbiota and aggregation in adults. We also investigated earlier about care, egg care and nymph care. So for this time, we provided them with the antibiotics, the females, for their entire life cycle, and we measure the effect on the expression of egg care. And surprise, surprise, there was also no effect. So the antibiotics did change the community of microbes in the gut of the females, but this did not translate into a change in the behavior. So overall, well, this shows that we clearly have no support for this very nice hypothesis, at least with the earwigs. And the point is that if you really think deeply about this new hypothesis, it's not completely surprising to not find any evidence, because actually the gut microbiota is, within the gut microbiota, there is a fierce competition between members to actually develop into the host. And so if one microbes develop the manipulative strategies, then it would pay a cost for developing and evolving these strategies, and the other will benefit from it, and they will basically be outcompeted by all the others. So there are some verbal arguments that are, in my opinion, very convincing, that shows that actually this kind of manipulation is very unlikely to evolve and occur in nature. Nevertheless, we still have two questions. The first one is whether actually the gut microbes of the host by itself is not very important for social evolution, but maybe the gut microbes of conspecific is important. It is known that the gut microbes will influence the chemical composition of other insects, and this chemical composition and chemical signatures is kind of important to drive social interaction. So maybe there is still a role for gut microbes in social evolution, but not the way we thought about it. And the last point is about the general importance of gut microbiota in earwigs per se, because basically what we showed earlier is that with the antimicrobiotics, there is no effect on the care, but there is also no effect on any other life history trait we have measured in the female. So now we are currently trying to develop axenic lines of the species to see whether indeed we can do it, and then to try to identify which component could be important for their life cycle. And with this, I will finish. I would like to thank all the funders that gave me money to test non-significant stuff, all the members of my group, and in particular, Marie-Charlotte Chutin, who is the postdoc who is currently working on the gut microbial stuff, and she needs lots of resilience. And also Manon Boussicot, who was at that time a master student who did the first experiment I presented earlier. And of course, I would like to thank you all for your attention. Thank you.
Is there any question in the room? Yeah.
Hi, thanks for the talk. It was very nice. Did you check if, because microbiota is efficient against parasites, so will it be like or like easy to check if you prioritized some earwigs and they will like transfer microbiota or like increase their microbiota to fight parasitism?
Yeah, that's a very good point, but actually never tested it. That's one of the plans. So I have a PhD student who is currently who just started a few weeks ago, and who is actually focusing on this kind of question. So one thing we plan to do is to actually manipulate the gut microbiota one way or another, and then to see and test whether there is an effect on the immune system by itself. So does it activate or not the immune system, and then to try to identify, if it does, which part of the gut microbiota could be involved in these things or not. So this link is something we plan to do in the future, but not for now.
Okay, now we have a question online by Thomas Lenormand.
Hello, thank you for your talk. I was wondering whether you looked at other things than bacteria in the gut. I'm thinking in particular to microsporidia. Actually, in my lab, we did some experiments on Artemia, and we found that those microsporidia did influence their aggregation level. So that's really something which is directly relevant to the question you were asking. Perhaps you know the paper. I can give you the reference. I gave the reference in the comments.
Excellent point. No, we didn't check for that, and I didn't know the paper, so I will read it carefully.
I will send it to you.
Thank you.
Was there a question? Finn?
I was wondering if you had in your for aggregation, did you have real controls without earwigs in the end? And what was the distribution? Because the distribution you have looks very much like autocorrelation with anything else.
I agree. There are some, I mean, this experiment took a lot of time. And once we finished it, we realized we could have done it. And that's clearly one thing we should have done. The other, to go deeper, another issue here is that we don't really know how it does change over time. So what, for practical reasons, we tested the aggregation with, for example, a group of 3 females in one set, and then we took the same test females several weeks later or several months later, and then we test the aggregation scores with other females. And we did, in some time, we did find correlation, and sometimes it was not correlated at all. And again, we don't know whether it's a seasonal issue or whether it's only driven by the group of attractant females. And this is something we plan to do this year, well, next year, to see whether actually we use the same kind of attractants, whether we always have the same level of aggregation. So whether basically the level of aggregation is driven by the host itself or by the conspecifics themselves, or whether it's completely random or at least driven by something else. But I fully agree. I mean, this random distribution would have been a good control for that.
Other question?
Like you say that earwigs parental care is non-obligatory. Is it experimental or also seen in the field?
That's a great question. The problem with earwigs is that we don't know much about its biology, in particular in the field, because they are nocturnal, which makes it quite difficult to observe in a natural condition. What we know is that in the field, we do see clutches without parents, without a mother. So we know that in nature, it's possible for the offspring to survive without, or to be without a mother for a long time, and to survive without a mother. What is sure is that most of our knowledge is based on laboratory rearing. And this is something that this is a major limitation here, because of course we have standard conditions and ideal conditions. And in such conditions, there are many species where family life becomes facultative as well. So I agree, this is a very good point.
Hello, I've known you for even longer. Slides where I wasn't really paying attention. What point of the microbiome? So you remove what they had and put something in, or you added?
No, again, so what we did is we simply added it. So, I mean, the point is that the fact that we do not have a strong correlation between the aggregation scores, the way we measured it, and gut microbiota somehow was a dead end for this experiment, but nevertheless we wanted to go further and just we tried. So we just gave them additional gut microbiota. So that means that indeed maybe the manipulation didn't work at all, which is likely according to the results. So this was just the first step, just to see whether as a matter of luck for once it could work without in a very rough way, and it's not the case. So of course it raises lots of questions.
And now a question online by Alice Maniel.
Hello, Joël. Thank you so much for your talk. It was super interesting. There is some publication in honeybees and bumblebees showing that, like individuals, like the new pup exposed to the feces of conspecific acquires a microbiota, and it's a necessary condition for them to then survive better to the exposure of a protozoa, like pathogens. So do you think in your species, it could be like saliva of the mother, for example, giving some microbiota strain to the pup, and this way, like this microbial exposure from the mother could explain the immune pattern later in life?
Yeah, well, thanks for the question. That's clearly an hypothesis we're working on at the moment. So now we're analyzing the data. So from Marie-Charlotte, so the postdoc, she did an experiment where we first follow the evolution of the gut microbiota throughout development. So through the different molts from the eggs to the adults, and see whether the molt by itself basically purge the microbiota or not. And the result is that it does not. And also we tested the role of maternal presence of the gut microbiota. So whether first instar larvae that remains with the mother, they do have a different microbiota if they have a mother or not. And then whether this is also the case 2 months later, whether they had a mother or not during the development. And we do find some differences. So the mother do transfer something that is present in the gut microbiota of the offspring. What they do transfer and how they do transfer is basically the next question. For now, we are just describing the phenotype, let's say. So yeah, but that might be very possible.
Okay, cool. Thank you.
I have because at the IRBI, as you know, there is some termite colonies.
I've heard about that. Yeah.
You try to give some guts of termite to your forficule.
What for?
Because the microbiota must be very different, and to see if there is an action of.
Well, the earwigs, they are omnivorous, and they're also highly predators. So they do eat other insects all the time. So basically, the kind of manipulation we did is something that they do in nature with many other insects. Whether they do, it does change their behavior or not. I mean, no, we never actually tested it, but yeah, that might be possible. I mean, there is no reason, that would be very surprising that just by eating another species, they would completely change such a major life history trait of them. But if yeah, why not? That's something that could be tested. I mean, indeed, there's plenty of earwigs that we want to get rid of, so I can do that.
Other question?
Thank you very much.